Hi, my name is Emma Schuette, and here is my presentation on reverse transcriptase PCR and COVID-19. So, COVID-19 is also known as SARS-CoV-2 virus, which is the scientific term for it. It is made of genetic material called RNA or ribonucleic acid. In our bodies, ribonucleic acid comes from DNA or deoxyribonucleic acid, which is that genetic material that's basically saying who we are and codes who we are. D sorry, DNA is transcribed into RNA, and then RNA is translated into protein, and that's how it is expressed through our body. That's known as the central dogma of biology. Um, Reverse transcriptase quantitative PCR is a method that is used to diagnose COVID-19. It is done by taking that sample, extracting that RNA or genetic material from the sample, reverse transcribing it into DNA, and then that DNA is amplified, so it's multiplied and multiplied and multiplied, and that gives a positive result because it shows that there is that RNA there, which means that there is COVID-19 present in the sample. So, like I said, the first step is that sample collection. That can occur through either your nose or your mouth or in the back of your throat or way back in your nasopharynx, which is like the very back of your nose, going down into your throat. That's not used as much now, and they use more the oral samples and the nose samples, and there's also rapid antigen testing, which is just another method of diagnosing COVID-19. The first step of our reverse transcriptase quantitative PCR is that reverse transcription. This part is where you are converting that RNA back into DNA. This DNA can be replicated just like it is in the normal body, so that's what's going to give you that amplification. More on that later. But as you can see here, we have the normal process of transcription where DNA is converted to RNA and RNA is then translated into protein. But you can also see the reverse there of the RNA into DNA. Basically, the primers have to prime that single-stranded RNA. It's only one strand when our normal DNA has two strands. So that one strand of RNA is primed so that it can be converted into that DNA, which is just like a copy of the RNA, but in its own terms. That's done through reverse transcriptase, which is an enzyme which basically pushes that reaction to occur or allows it to occur. And then ribonuclease degrades and removes the RNA strand so that the DNA can double strand and then you'll have the DNA to replicate. The next step is the quantitative PCR. Quantitative refers to the fact that we are measuring it and giving a numerical value. We're measuring the amount of DNA once we amplify it. And then PCR actually stands for polymerase chain reaction. The very first step of PCR is to denature the sample, and that's the DNA sample. It's done at 95 degrees Celsius, which is a very hot temperature. This removes any RNA, and then annealing lowers the temperature to about 60 degrees Celsius, warm but not warm or hot, but not as hot as 95 degrees Celsius, obviously. And this allows for the DNA to bind to a fluorescent probe of that will emit light, and then the machine, as you can see here on the right, will be able to measure that light. The last step is extension or elongation. This is where that amplification occurs of the DNA, and that's done with Taq polymerase, which is another enzyme, so that allows that reaction to occur. Taq polymerase can only amplify DNA, which is the reason that we have to convert the RNA into DNA to be able to measure it or quantify it. So as you can see here, this is an example from an article that describes this process. On the left, we have an RNA sample, and on the right, we have a no RNA sample. So basically, if this was a COVID test, on the left, we have a positive COVID sample, and on the right, we have a negative COVID sample. So as you can see in that positive COVID sample, there is that RNA present. So the RNA is reverse transcribed into that DNA, and then that DNA is denatured and then annealed, and that will give the DNA will break apart so that the probe can attach. And then when that probe attaches, then we can measure it and amplify it and then measure it, and that will give us our positive result. On the right, you can see that there was no RNA, so there was no reverse transcription, so there is no genetic material for that Taq polymerase to amplify. So that shows us on the bottom. The left side would give us that green result or the positive result where it's amplified and there's multiple of that sample, while the negative result is going to remain a baseline. It's not going to amplify, which means that you didn't have COVID because you didn't have that RNA in your mouth swab or your nose swab that would give you that result. And then here are my references. Thank you.